Hi, everyone. I'm just going to share my screen here. Okay. So thank you all for coming and thank you for the invitation to speak here. My name is Abhay Hukku. I am a current PhD student in the Department of Biostatistics, and today I'm going to be talking about BAGSE, which is a Bayesian hierarchical model that set enrichment analysis. So at any time today, if you have any questions about anything I talk about, please just unmute and feel free to interrupt me whenever. And yeah. So just as a general outline for this talk, first I'll be talking about the motivation and background behind conducting gene set enrichment analysis in general. I'll then talk about some of the existing methods for gene set enrichment analysis and their limitations. I'll then introduce BAGSE and some of its features and how we try to address those limitations. And I'll go on and show BAGSE's performance in simulations and real data applications before wrapping up with some conclusions. So oftentimes the results of a GWAS or differential expression analysis can give a very long list of seemingly biologically unrelated significant SNPs or genes. And this is especially true over time as sample sizes have increased. We end up with many signals of significant SNPs or genes from these studies. And although having this many significant results is great, it can become increasingly difficult to find any sort of unifying biological theme to link these results. And so it becomes hard to subsequently use these results to gain more insights into biological mechanisms behind diseases. And so there has become a need to develop techniques to extract more biological meaning from the results from these analyses to help to uncover these sort of biological causal mechanisms behind diseases. And one such technique is known as gene set enrichment analysis. So gene set enrichment analysis is performed downstream of a differential expression analysis or a GWAS. And the goal is to identify groups of genes that have a higher or a lower number of association signals than expected. So oftentimes gene sets are defined as genes in the same biological pathway. And in this way, it can give more biologically meaningful results because now instead of saying we have these genes that are associated with this disease, we can say genes involved with this biological function are associated with this phenotype or disease. And so this can gain more insights to subsequently do further research into seeing maybe how that specific biological function could be connected with the disease. And in this way, gene set enrichment analysis was first developed in 2003 by Mutha et al. And they found that genes involved in oxidative phosphorylation had lower expression in human type 2 diabetic muscle. And so this is just one way gene set enrichment analysis has been used to gain more biological insights into certain diseases. So one way that we believe is a natural way to think about gene set enrichment analysis is through this latent 2x2 contingency table interpretation. So each gene in a study is either unannotated or not in the gene set or annotated in the gene set. And the placement of the classification of genes as in the gene set or not is going to be determined prior to the study by the investigator or researcher who classifies each gene as in the gene set of interest or not. Additionally, each gene is either associated with the phenotype of interest or not associated with the phenotype of interest. And in a differential expression setting, this would also translate to each gene as either differentially expressed or not differentially expressed. So thinking about it this way, each gene can be mutually exclusively classified into one of these 4 cells. And so we can create this 2x2 contingency table. And when this table is developed, we can then easily calculate an odds ratio. And this odds ratio falls very well in line with our definition of enrichment because the literal interpretation of it is a gene that is annotated has OR times higher odds of being associated with the phenotype of interest than a gene that is not annotated. Because of this, and because odds ratios are used so prevalently throughout statistics in biology, we believe that this value is the ideal quantification of enrichment, and it will be the value that we're estimating with our method. So because we believe this 2x2 contingency table is a very natural way to think about enrichment, I will be referencing this throughout the talk, especially when explaining our method. So please keep it in mind, and I will reference this again. So for the existing methods for gene set enrichment analysis, they typically fall into two different categories. The first category is GSEA-based methods for gene set enrichment analysis. So in this school of method, first association scores are assigned for each gene. And this association score can be a variety of different things. It can be a z-score, it could be a p-value, or it could be just an effect size estimate, like estimated differential expression. And then within the GSEA-based methods, there's the unweighted GSEA-based approach, where the distribution of these association scores are compared between genes in the gene set and genes not in the gene set using a statistical test such as a Kolmogorov-Smirnov test or a Mann-Whitney-U test. And then there's the weighted GSEA-based test, where you first generate a ranked list of genes by that association score, and then you calculate a running sum statistic down the list. So starting at the top, you go down the list, and when you run into a gene in the gene set of interest, you go up by a certain weight, and then when you run to a gene not in the gene set of interest, you go down a certain weight. And in this procedure, you then use permutations to obtain an empirical p-value. Oh, sorry, and the enrichment score is the maximum deviation from zero while calculating this running sum statistic. And some of the approaches that use this type of method are gene ontology, GSEA, and MAGMA. So the second category of gene set enrichment type methods is the two-stage procedure. So the first stage in this two-stage procedure is known as a classification stage. And here, each gene is first classified as annotated or unannotated as defined by the investigator, and then classified as either associated or unassociated through different types of methods, such as p-values using a Bonferroni correction, or q-values, or a p-value threshold. And of course, once this classification stage is over, each gene can be assigned to one of these four cells in this contingency table. And once this assignment is done, some sort of statistical test can be used to calculate if there is significant enrichment, such as a chi-square test, a Fisher's exact test, or another permutations-based test. And some of the approaches that use this type of method are David, Enrich, and Magenta. So one thing to note is that using a two-stage procedure, you can get this ideal odds ratio of enrichment because you have generated this 2x2 contingency table. So some of the limitations within these existing methods. So first, for the GSEA-based methods, the permutations-based procedure for the weighted version to obtain an empirical p-value is not exact, and it can also be computationally expensive, especially as the number of genes being investigated increases. Doing a permutations-based procedure to calculate a p-value can end up taking a lot of computational power and time. And also, there's no easily interpretable value quantifying enrichment that can be used to compare gene sets. So there's no odds ratio that we had talked about earlier. And then for the two-stage methods, the uncertainty that's introduced in the classification stage, specifically the uncertainty introduced when classifying the genes as associated or unassociated, is then ignored in stage 2, the testing stage. So although type 1 error for the classification stage can be accounted for through type 1 error correction methods such as Bonferroni correction or using Benjamini-Hochberg approach, the type 2 error introduced is not necessarily accounted for. And so to address these concerns, we developed BAGSE, Bayesian analysis of gene set enrichment. And so we developed a Bayesian hierarchical model for gene set enrichment analysis that can be run using only summary statistics, so only z-scores or beta hat and standard errors from a differential expression analysis or GWAS. And we quantify enrichment as the log odds ratio of interest that we talked about, which we call alpha 1 in our model, which I'll explain further in the next slide. And a couple other interesting features about BAGSE. The enrichment that we calculate can then be used as prior information to improve power of gene level testing, taking advantage of an empirical Bayesian framework. And also BAGSE is adaptable to testing multiple overlapping gene sets. So here is an overview of the Bayesian hierarchical model we developed. So on the left here is a graphical model representation of this Bayesian hierarchical model. So the first level is gamma I, which is the binary association status of gene I. And the distribution of gamma I is impacted by the enrichment parameters alpha 0 and alpha 1, which we have to estimate, and also impacted by DI, which is the binary annotation of gene I. And I should say that this model that we're looking at here is for when we are looking at one gene set at a time. So DI is either 0 or 1. And so the prior distribution of gamma I here is a Bernoulli distribution that with the parameter impacted by these enrichment parameters and DI. And then for the next level of the hierarchical model, we have the true effect size of gene I. So this true effect size is conditional on gamma I in that when gamma I is 0, that means gene I is not associated with the phenotype, and the true effect size of gene I will be 0 and not have a distribution. It will just be a point mass at 0. However, when gamma I is 1, then beta I follows this mixture normal distribution with k components. And one thing to note is that this number k, so the number of components, and phi, which is the variance of each component, is estimated in a data adaptive way proposed by Matthew Stevens in a 2016 paper. And this pi, which is the probability of being drawn from each of those k components, is a parameter that we have to estimate. And one thing to note is that the pi parameters actually is different dependent on whether the gene is in the gene set of interest or not. So that is one of the unique statistical capabilities of our model, in that the prior distribution for the true effect size is not going to be the same across all genes. It'll be different depending on if the gene is in the gene set of interest or not. And then for the third level, we have the observed estimated effect size beta hat I, which is conditional on beta I, and this distribution is just normal with mean beta I and a standard error that we can estimate. So one interesting thing to note here, looking at the prior distribution of gamma I, is that this distribution is exactly like how the distribution for an outcome of a logistic regression, the outcome variable for a logistic regression would look like, and with DI being the predictor variable. So when you think of it like that, it becomes more clear how alpha 1 is that log odds ratio of enrichment, And the only difference here is that in a real logistic regression setting, the outcome variable will be observed, whereas here, of course, gamma I, the true association status of gene I, is not actually observed. So because of this, to estimate maximum likelihood estimators for alpha 0 and alpha 1, we used an expectation maximization algorithm, treating gamma as missing data. But before I go on, I want to pause for a bit because I know this slide is very involved with sort of the meat of our whole methods. So I want to, in case there are any questions or anything is unclear. Okay, if there's no further questions, I will go on to explain an intuitive explanation behind this EM algorithm that we use to estimate the enrichment parameters. So here we use this EM algorithm to estimate alpha 0 and alpha 1 and also pi, but I won't really be going into the estimation of the pi parameters because they aren't as relevant as the enrichment parameters for this talk, and they're not as intuitive and a little more computationally complex. So here I'm going to be just explaining how we calculate alpha 0 and alpha 1. So first we initialize all the parameters that we need to estimate. And then in the E step, we have to find an expectation for the missing data. So here we end up having to find the expectation of the indicator that gamma I equals 1, conditional on all the observed data, like the beta hats and their standard errors, and the annotation of the gene, and also our current parameter estimates of alpha and pi. So this term is actually statistically equivalent to the probability that gamma I is equal to 1, conditional on all the observed data and the parameter estimates. So if you can think about this again in a latent 2x2 contingency table setting, this actually can be used to basically fill out iteratively this 2x2 contingency table. So let's just say gene I is in the gene set of interest, so it is annotated, and we calculate this probability to be 0.8. So then we would fill in we would add 0.8 to N11 and 0.2 to N10. And we do this for every single gene for each iteration to calculate a teeth iteration of this whole 2x2 contingency table. And you can see how, contrary to the two-stage approach, which each gene is put in as a 1 or 0 in one of these 4 cells, we incorporate the uncertainty and insert this probability into into a couple of these 4 cells. So then once this 2x2 contingency table has been developed for an iteration, the M step can be used just as calculating the log odds and, of course, alpha 1, the log odds ratio. And this will end up being the maximum likelihood estimate for alpha 1, which can then be used back into the E step. So this is a natural way to see how our approach in BAGSE incorporates the uncertainty from classification, whereas the two-stage approach does not. And so we can do this EM algorithm until convergence and obtain the maximum likelihood estimates for the enrichment parameters. And we can easily get standard errors for these parameters also through a profile likelihood approach. So one other unique feature of our method is that the enrichment information that we calculate can then be used as priors using a parametric empirical Bayesian framework outlined by Matthew Stevens. And it can be used to boost the power of testing the hypothesis that gamma I is equal to 0, which means gene I is not associated with the phenotype. So we do this by calculating this local false discovery rate for each gene. And this local false discovery rate is equal to the probability that the null hypothesis is true given all the parameter estimates and the observed data, which is here equal to the probability that gamma I is equal to 0. And one interesting thing to note is that this probability is actually just 1 minus this probability from the E step. And so we actually already have this quantity from that EM algorithm. So no extra computational work is required to get these local false discovery rates for every single gene because we just use from the last iteration in the EM algorithm for every single gene. And then this local false discovery rate can subsequently be used for gene level testing using a standard Bayesian false discovery rate procedure. So now I want to give a brief overview of how BAGSE can extend from one gene set to multiple gene sets. So let's assume we have L gene sets that we are simultaneously investigating. So now DI, instead of just being a 0 or a 1, it is an L-length vector where each component is either 0 or 1. So this allows for overlap of gene sets because you can have multiple ones within this vector for each gene. And each different configuration of the vector, each possible different configuration of the vector has its own associated enrichment parameter. So one of the drawbacks to this procedure is that we end up with 2 to the L enrichment parameters, which you can see can become very computationally expensive as L increases. However, one of the benefits to this approach is that it maximizes the gene level discovery power for the incorporation of enrichment information to gene level testing, which makes intuitive sense because the more information that you're gaining about the gene, the more power you will have to discover if the gene is associated or not. Okay, so now I will get into the simulations where we talk about the performance of BAGSE. So here is an overview of the simulation scheme that we use. So for each simulation, we simulate 10,000 genes, and the proportion of genes annotated varies between 1% and 20%. And the proportion of genes annotated does have a very profound effect on the power and stability, which is like the variance of the enrichment estimate. But the effect it has on all methods is around the same. And so for each gene, we simulate a z-score. For null genes that are not truly associated with the phenotype, we generate their z-score from a standard normal distribution. And for associated genes, we generate their z-scores from a t-distribution with 10 degrees of freedom. And the number of associated genes depends on the enrichment parameters. So basically, for each gene, we already have whether it is in the gene set of interest or not. And then we use the enrichment parameters and the annotation information to generate from this Bernoulli distribution to classify it as either truly associated with the phenotype or not associated with the phenotype. And so about these enrichment parameters, alpha 0, we set to -1 throughout simulations. And this value is just from what we observed in real data. The log odds of a gene being associated generally seems to be around -1. And then we vary alpha 1 between 0 and 1 for these simulations. And for each parameter set, we conduct 5,000 simulations. So this is the general simulation scheme we use. We make a couple minor changes for some of the individual simulations that I'll mention when we get there, but in general, this is the scheme we used in our evaluation of BAGSE. So the first thing we looked at was a closer look into the limitations of the two-stage approach. So here we generate data only using an alpha 1 of 1, and a proportion of genes annotated of 10%. And we just look at how the gene level power, which is basically the power to classify a gene as associated in stage one of the two-stage approach, and how that impacts the subsequent estimate of the log odds ratio of enrichment. And we see that, as expected, as gene level power is very low or close to zero, there's severe underestimation of the log odds ratio of enrichment. Whereas when the power gets close to 1 to detect an associated gene, the underestimation is much smaller and is very close to the true estimate. Works to help confirm our hypothesis that the two-stage approach, the ignoring of type 2 error in the first stage does impact the subsequent estimate of enrichment for the gene set. So now we looked at simulations to compare this two-stage approach with BAGSE in terms of enrichment quantification. So this graph is looking at the results for when 10% of genes were annotated, and we generate data from these 5 different true enrichment parameter values. And we see the pattern we would expect in that at low levels of enrichment, both are very close to the true value, both BAGSE and the two-stage approach. But then as the true level of enrichment in the data grows higher, this two-stage approach starts to severely underestimate the true enrichment parameter, while BAGSE seems to maintain a mostly unbiased estimate of the enrichment. We see here a slight overestimation, but this doesn't seem to be a pattern, as we also looked at higher levels of enrichment, and this amount of overestimation was around what we saw. And so, yes, this confirms what we saw in the last simulation, that the two-stage approach at higher levels of enrichment tends to underestimate the log odds ratio of enrichment. So then we looked at the power to detect enrichment. And again, here we looked at just 10% of genes annotated. And so for this, we also included weighted and unweighted GSEA in our method comparisons. So one thing to note is that when the true enrichment parameter is 0, this value is actually the type 1 error rate. And we see that all methods do control the type 1 error rate reasonably well at around the 5% level. And we see that for the medium enrichment parameters of around 0.1, 0.25, BAGSE and weighted GSEA do seem to overperform the other two methods. And then once the enrichment parameter is around 0.5, all of these methods have a very close to 100% power in detecting enrichment. And so we believe that in terms of power, once the enrichment level is at a reasonable level in real data, all methods do have very good power in detecting the enrichment. So now we decide to look at the incorporation of the enrichment information in subsequent gene level testing to see how that impacts the power of gene level testing. And so here we use the same simulation scheme, but we extend the alpha-1 parameter set in order to see how the pattern will evolve. And so we saw that, so for this graph, the y-axis is the percentage of power increment that we gain using BAGSE's method to incorporate enrichment information from these two approaches, which is a Q-value procedure and compared to a local false discovery rate procedure that does not include enrichment information. And we see that at low levels of enrichment, which is like we were looking at with 0.5 and 1, there's a very modest power boost that we gain in gene level testing, maybe around just 2%. But then as the true enrichment parameter grows higher, and especially as it grows very high, like in the 4 or 5 range, the incorporation of enrichment information can give a pretty substantial boost to the ability to detect gene level associations of near 20% from a Q-value procedure and from a local false discovery rate procedure. So now I would like to talk about our real data applications of BAGSE. So firstly, we applied BAGSE to this differential expression analysis done in Moyer-Brillian et al. In 2016, where they conducted differential expression analysis of 20,896 genes under Cook's analysis. We got p-values and beta hat estimates from DCMB. So from this information, we were also able to generate standard errors of the beta hat, which we could then use in our analysis because BAGSE only requires summary statistics to be used. And through our conversations with collaborators, we expected genes and pathways corresponding with immune response to be enriched looking at this data set. And we saw using the KEGG database that 21 pathways were involved in immune response. And so because we weren't exactly sure which of those pathways should be associated, should be implicated in this analysis, and each of these pathways had a very small number of genes present in our data set, we ended up aggregating all the genes in these pathways for our analysis. So what that means is that the annotation for each gene was 1 if it was in any of those 21 pathways, and it was 0 if it was not in any of those 21 pathways. So using this procedure, we have 2.7% of genes that ended up being annotated. And then we ran all 4 methods on the data and saw each of them detected significant enrichment, while BAGSE estimated the alpha-1 enrichment level to be 1.31, and the 2-stage approach had a 0.87 estimate of enrichment. So this difference in enrichment estimates does fall in line with our simulation results, showing that the 2-stage approach may have underestimated the enrichment level. And at this level of enrichment, it makes sense that all 4 methods should have 100% power to detect the enrichment. So we also incorporated this enrichment information to see if we could discover more genes in this gene level testing. And we see that compared to a local false discovery rate, using enrichment information, we can discover 90 more genes in the analysis. So we also applied our method to a transcriptome-wide association study. So a TWAS is a type of study that integrates eQTL and GWAS to study relationships between the expression of candidate genes to the complex trait of interest. And the way it does this is by first training a predictive model of gene expression using the eQTL data sets, and then applying that predictive model to the GWAS. So you're using that model to impute gene expressions in the GWAS data, and then a TWAS looks at the gene expression and complex trait associations to generate z-scores on the gene level. So we get GWAS summary statistics of HDL levels from the Global Lipids Genetics Consortium, and we also use multi-tissue eQTL from the GTEC project. And to conduct the TWAS, we use S-PredictScan to obtain TWAS z-scores and p-values for 32,362 genes. And so this was our way of getting gene level statistics from the GWAS. So rather than using a method such as a proximity-based approach to get the gene level statistics, we used the TWAS. And we compared our results of running gene set enrichment analysis on this data to those from GLGC, which used magenta, which I had mentioned is similar to a two-stage type of approach. And they used magenta for the gene set enrichment analysis. So we looked at 6 gene sets that were implicated in the GLGC paper, and we analyzed them using BAGSE and weighted GSEA. So we see that looking at the actual point estimates of enrichment we get from BAGSE, there does seem to be enrichment in each of these different pathways, although looking at the p-values, the p-values may not be as super significant as we might expect, seeing such high levels of enrichment. And we think this is because TL study, the sample size isn't as large, so that makes the TWAS lose a little bit of power, which may have affected our subsequent analyses. But we still think that the fact that we were able to see some all point estimates being above 1 is still a promising finding. And we mostly wanted to demonstrate the ability to apply BAGSE on this type of data set, as we think that a TWAS is a growing type of data used to get gene level statistics from a GWAS study. And again, we used the additional gene discovery feature of BAGSE here to see how many more genes we can discover. So using a local false discovery rate with no enrichment information, there were 578 genes discovered. Using the cholesterol metabolic process level of enrichment, which had the highest point estimate of enrichment, and using that as prior information, we discovered 30 more genes. And then if we use all 3 Go terms enrichment as prior information, so doing using multiple gene sets to conduct enrichment analysis, and then using that as prior information, we discover another 18 genes. So that just gives a bit of insight as to how this approach can be used to aid in gene discovery. So now in conclusion, our proposed method BAGSE gives an easily interpretable and accurate measure of enrichment for gene sets that maintains the power to detect enrichment from the state-of-the-art methods. Also, BAGSE presents a framework to naturally improve power in gene level testing using enrichment as prior information. And also, BAGSE can be naturally applied to TWAS, which is a growing data type that integrates EQTL and GWAS. So for more details, we have this paper published in Bioinformatics, and we have our software at this GitHub of William Len. And the software is implemented in C++, so it'd be used in the command line. And yeah, thank you to William, Corbin Quick, Roger P. Griggy, Francesca Luca, and GSTP for funding this work. And thank you to all of you for listening. And I'd be happy to take any questions now.
Thanks for the interesting method. Thanks for the talk. One of the applications of the gene set enrichment is as a retrospective analysis for a new data source to say if it's interesting for determining new gene functions. But in this case, part of the assumption is that there's new gene functions to annotate, to find. And so built into this application is the idea that the annotations are somehow incomplete, that you have, that there's, like say in the Go term annotations, that there are some genes that are annotated for it and some genes that are not annotated, and some of those are missing. And I was curious if you could, how you would incorporate that assumption or that prior, or how that would fit into your model for the idea that there's, that the annotations are there that are good, but then there could be missing annotations. So,
okay. So you're saying that in a certain study where many genes are annotated, but there's also a lot of genes not annotated that should be annotated.
That's right.
Okay.
So rather than just the binary annotated, not annotated, you have kind of a missing or,
I see.
One positive and unannotated versus positive and negative.
I see. Yeah, so that's not something we actually considered in this model, and that would definitely be something interesting to look at because, yeah, in this model, we assume that annotation is known as being classified in the gene set. So that would definitely be an interesting future direction that we should definitely look into to maybe not dichotomize the annotation for each gene. I think that would be interesting. Yeah, yeah, that would be interesting to think about. Thank you. Any other questions?